Alright, so now we're going to move on to carbohydrates. So carbohydrates serve as fuel and building material for different organisms. So carbohydrates include the sugars and the polymers of sugars, so single and multiple sugars joined together. The simplest carbohydrates are monosaccharides or a single sugar. And carbohydrate macromolecules are polysaccharides, and these are composed of many sugar building blocks. So monosaccharide is one sugar, and then polysaccharides is many sugars. Alright, monosaccharides usually have molecular formulas that are multiple of CH2O, so carbon, hydrogen, and oxygen. So glucose is the most common monosaccharide, and its formula is C6H12O6, and you guys should know this. This one's important. Monosaccharides are classified by the location of the carbonyl group and the number of carbons in the carbon skeleton. So what we mean by this is, as you can see in the orange rectangle here, the C and the O, the carbon and the oxygen, that is the carbonyl group. And then, so you can see it can either be an aldose or a ketose. So an aldose has the carbonyl group at the end of the polysaccharide, while a ketose has it in the middle, or at least within the polysaccharide, or at least, yeah. So aldose outside, ketose inside. And then another classifying aspect is the number of carbons in the carbon skeleton. So you can have multiple carbons in the skeleton. So for example, in a triose, you have 3 carbons. Pentose is 5. Hexose has 6. So two different characteristics is location of carbonyl group, number of carbons. Here's a little more detailed picture. And so even though we often draw these structures as linear skeletons, when they're in aqueous solution, so we talked about that earlier with water, many sugars form rings when they're in water. So monosaccharides serve as a major fuel for cells and as raw material for building molecules. So here we go from a linear form to a ring form. So here's what the ring form looks like. Here's a more detailed. Alright, so a disaccharide is formed when you have a dehydration reaction, so taking out water to join two things together. So dehydration reaction joins two monosaccharides. And this covalent bond is called a glycosidic linkage. So here's a couple examples of joining two glucose monomers together, taking out the water to form a disaccharide, and that one's going to be called maltose. And then here's another example of glucose and fructose. Remove the water there, and now you have sucrose. So I'm sure you guys recognize all these names. At least you've seen the names before. So that's what they are. That's how you get them. Alright, so polysaccharides, monomers of sugars, they have, like we said, storage and structural roles. So what are the two roles of polysaccharides? It says storage and structural roles. Alright, so the structure and the function of a polysaccharide are determined by its sugar monomers and the positions of its glycosidic linkages. So the actual individual's monomers within it and how they're joined together. Okay, so for example, starch. And starch is a storage polysaccharide of plants, and it consists entirely of glucose monomers. So plants store a surplus of starch as granules within chloroplasts and other plastids. So you guys have seen chloroplasts before, little green discs in plants, and so this is where they're going to store starch. However, conversely, in animals, you can see to the right, they have glycogen as storage. So glycogen is storage polysaccharide in animals, and other animals and other vertebrates store glycogen mainly in liver and muscle cells. So just as a side note, if you see starch in plants, they're kind of like linear, maybe a few linkages like amylose doesn't have that many little branches off of it. Amylopectin has a few, but glycogen in animals need a lot more energy, so since these polysaccharides store energy, how you can see there's a bunch of branches in there. So if you can kind of picture, you like take your hand and kind of spread it out, and then like point to the middle of your the palm of your hand. It's kind of shaped a little bit like that. Like it has like a central point, and then it has like fingers coming off of it of these strands of polysaccharides. And so how you get energy from these polysaccharides is that enzymes come and they eat off essentially each monomer, each monosaccharide. It eats it down and utilizes it for energy. However, with animals, you want to get as much energy as possible you want to get as quickly as possible. So if you have lots of branches or lots of points of entry, if you will, to be able to eat that down, you can eat that down a lot more quickly. So that's as opposed to just eating one down at a time. So it's like having a multi-lane highway. And as for glycogen, get a bunch of cars going, or eating a bunch of those sugars down at a time to get lots of energy in an animal, as opposed to just like a single or double lane highway in a plant. Just need it as quickly. Anyways. Okay, now we're going to go to cellulose, which is a structural polysaccharide. So here we're talking about storage, like energy, sugar, and now we're talking about structure. So for cellulose is an example of a structural polysaccharide. And so cellulose is a major component of the tough wall of plant cells, and you guys have probably heard of this before. So like starch, cellulose is a polymer of glucose, but the glycosidic linkages, or how they're joined together, they're different. So the difference is based on two ring forms of glucose. So this is getting a little complicated, but don't worry, you guys can get it. So the two different types are alpha and beta linkages. So for some reason the alpha and beta are not showing up in here, but the blue ones are alpha and the gray ones are beta. So if you guys can see, remember when you saw the dehydration reactions, you saw how the rings were joined together. So, and then you also see, so that's one component, but then the other component is, see the little OH that's highlighted in yellow? That is one way that, when you're just looking at the orientation of glucose, when you join them together, you can either join them together with the glucose having all of the OHs down on the same side, so that is an alpha linkage when they join together, so that the glucose's OH group is pointing down and all on the same side. But if you can also link them, you can flip one of the glucoses, so have one OH pointing down and one pointing up, and link them together like that, so every one is opposite, and that one's called a beta linkage. So not too difficult when you join together, just depends which orientation the OH group on the glucose is pointing up or down. So here's alpha and beta. So you can see, so it just depends. You can link together, you can still link together glucoses, but if you link them together in an alpha way, they're all the same monomer, they're all the same Lego brick, if you will, kind of, but if you link them all together with the glucose's OH group pointing in the same direction, you get starch, but if you point them in alternating directions, you get cellulose. So the way you join these together is going to affect the shape of the chain. So like polymers with an alpha glucose, so the OH group's on the same side, they'll end up being helical. And then polymers with the beta-glucose linkages are going to be straight. So in straight structures, the hydrogen atoms on one strand can bond with the OH groups on the other strands. But in parallel cellulose molecules, they're held together and grouped into microfibrils, and those build strong building materials for plants. So here's an example of cellulose fibers in plant cell walls, so you can kind of see how they're structured together and how they're built of cellulose molecules, like all grouped together in a microfibril. Okay. Alright, so enzymes that can't digest starch by hydrolyzing the alpha linkages, or that hydrolyze the alpha linkages cannot hydrolyze the beta linkages in cellulose. So in other words, the enzymes that break down a starch can't break down cellulose, and that's because of the way those two monomers are joined together. So the enzyme is a certain shape that only breaks down the alpha linkage, but it can't do the beta linkages. So cellulose in human food just passes through the digestive tract as insoluble fiber. So you guys probably know, like, to eat your vegetables and like get enough fiber. And one reason why you want fiber is just because it helps things go on through, and it goes on through because it's not broken down. And it's not broken down because humans break down starch and not necessarily cellulose. So those enzymes can't break down cellulose, and things just keep passing on through. But some microbes use enzymes to digest cellulose, but those are mostly in herbivores like cows or termites, and they have symbiotic relationships with these microbes, so bacteria in their gut helps break those things down. So you need certain enzymes to break down these linkages, and humans don't necessarily have them for cellulose, but other animals that are strictly herbivores do. Here's an example of a cow with a bacteria, a microbe, and the microbe has those enzymes that break down the grass that it eats, that contains cellulose. So that makes sense? All right, another structural polysaccharide is called chitin, and is usually found in the exoskeleton of arthropods, so like lobsters or crawdads, or something like, those arthropods with the hard exoskeleton. And so these also provide structural support for the walls of many fungi as well. So here's an example of structure of a chitin monomer. Here is like a cicada that is an arthropod shedding its shell, so that's all made of chitin. And then chitin is also used to make surgical thread. Alright, and so that is the end of this lecture about carbohydrates.